Hello, and welcome to this week's Frontiers in Oncology, where we have a special guest speaker, Scarlett Lin Gomez, and she is going to be presenting on multi-level studies to address cancer health disparities from omics to structural and social determinants of health. Dr. Gomez is a professor in the Department of Epidemiology and Biostatistics and a member of the Helen Diller Comprehensive Cancer Center at UCSF. She is the co-leader of the Cancer Control Program and a member of the Prostate Cancer Program Leadership Committee. She is director of the Greater Bay Area Cancer Registry, a participant in the NCI SEER program and the California Cancer Registry. Dr. Gomez was an SCI member for a decade before joining UCSF and has been an amazing collaborator with many SCI members, including myself, where we have many publications in lung cancer. Her research focuses primarily on cancer health disparities and equities and aims to understand the multi-level drivers of these disparities. I'm really excited to hear her talk today. So welcome, Dr. Gomez.
Thank you, Dr. Wakely, and I'm so honored to have been invited to share some of our work from our lab with this group today. I'm going to go ahead and start my slides here. So I'm going to be sharing some of our work that focus on trying to combine looking at factors ranging from omics to structural and social determinants of health to conduct multi-level studies that address cancer health disparities. This backdrop that you see here actually sets a nice stage for for my talk. This is one of the standard PowerPoint templates that we're required to use for UCSF. And what I think is really notable about this, it's actually an aerial view of Mission Bay. This is the campus that one of the sites that my group resides in. This is Mission Hall right here. We're on the second floor of Mission Hall. And in fact, you already can tell that if you are familiar with this area of San Francisco, that this photo is already dated. This parking lot here actually is a new neuroscience building that was just inaugurated, I believe, a couple weeks ago. And this empty space here is actually now the Warriors Stadium. So what I wanted to point out here is that this street here is 3rd Street. This is Mission Bay, obviously thriving, commercial, live, work, campus, and lots of different industries in addition to UCSF, obviously. If you travel down just about a mile, you reach the neighborhood called Dogpatch, which really is a transitioning neighborhood. We go there often because they have a lot of cool eateries. My daughter goes climbing there, you know, sort of hipster, if you will, type of neighborhood, young working adults. If you travel down another half a mile, you reach a neighborhood called Bayview Hunters Point. Historic shipyard, primarily Black and brown populations living there. If you look at the census demographics, much lower socioeconomic status profiles, much higher percentage living in poverty, as well as unhoused populations. So I wanted to illustrate this because just even within our own city that we live in, we see the remnants of structural racism over the years. This arose as a result of redlining, and that has endured to this day. So I'm going to talk a bit about how we conduct research to address how these historical structural factors may have impacts on the current health disparities that we see today in cancer. I like to start with this quote. This is from social epidemiologist David Williams. This was from the PBS documentary Now, quite a number of years ago, obviously in 2008, but many of the messages and themes that were presented in this documentary are certainly still relevant today. And I'd encourage you, if you haven't yet, to go onto the PBS website and stream this. It's still available, and you can also read the transcripts. So I particularly like this quote to sort of set the stage for our work. He talks about, when we typically think about health, we think about certainly healthcare, access to care, quality of care, but what research clearly shows us that health is embedded in the larger conditions in which we live and work. So what implications does this have for intervention? Sometimes we naively think of improving health by simply changing behaviors, but the choices of individuals are often limited by the environments in which they live. So if we don't address the environment, how can we address people's health behaviors and health outcomes? And in fact, there's a continuum of influences on health outcomes. Not only is there a continuum, there's these continuums have an explicit ordering of influence. In fact, there's the upstream factors and the downstream factors. Downstream are the consequences on behaviors and health. Upstream are factors that we typically are starting to hear a lot now, especially in research, even in the social media, certainly in the past year during living through this pandemic, about the social determinants of health, which the WHO defines as the conditions in which people are born, grow, work, live, and age, and the wider set of forces and systems shaping the conditions of daily life. So these are the conditions: the physical environment, economic, service environment, and the social environment in which we live, work, live, and age. But what about the structural determinants of health? These are the upstream determinants, the social inequities as determined by who we are, class, race, ethnicity, immigration status, gender, sexual orientation, and the intersection of these statuses, as well as institutional inequities, policies, programs, and practices within different institutions that drive who receives services, who gets to receive services, who gets to live where they live. So kind of trying to apply that framework, we try to apply a multi-level approach in the research that we do, focusing on structural and social determinants, those upstream factors. We use this framework that was developed called the Celsus Society model for health disparities research. This was developed by a group funded through a series of P50 grants called the Centers for Population Health and Health Disparities at the NIH. And what it does is it posits that, or frames the multi-level factors in a vertical framework, ranging from here at the bottom, we see the biological factors, genetic pathways, biological responses, to individual level factors. We move up the framework, social and physical context. And then again, we see these fundamental causes in terms of structural context and institutional context. And importantly, this framework, this model emphasizes the interactions across these factors to impact cancer health disparities. So we keep this in mind as we design studies to address cancer disparities. So one example that I like to present in which we try to utilize this framework is our portfolio of work looking at lung cancer in Asian American females who've never smoked. And I'm glad to be able to present this work to you today, as it's actually it reflects a long, going on a decade now of collaboration with Dr. Wakely. Why lung cancer in Asian American females? We know that the burden, that lung cancer is a leading cause of cancer mortality among all Asian American populations. This was a publication we did in CEBP a few years ago. And what it shows here are the top percent contributors, the rank based on the age-adjusted mortality and the percent of contributors to cancer deaths, the top 5 cancer sites for each Asian American ethnic group shown here, the top 6 shown here for males and for females. And so what you see here for females is for these top 6 largest Asian American ethnic groups in the US, lung cancer ranks as the number 1 or number 2 for among Asian Americans, very closely ranks as the number 3 contributor to cancer mortality. Why is this notable? Well, this is notable because if you look at population level risk factor data, the smoking prevalence for most of these Asian American ethnic groups among females is exceedingly low, typically less than 10%. And in fact, we know that this is a diverse population, the Asian American, Native Hawaiian, and Pacific Islander group. Along with Dr. Wakely, we've been publishing based on cancer surveillance data and others, documenting that lung cancer incidence varies widely across these populations, these groups. We know from looking at case series data among those diagnosed with lung cancer that more than 50% of cases among Asian American, Native Hawaiian, Pacific Islander females are never smokers. This is much higher in contrast to what we see in the general population of US females, about 15%. And until now, we've had no sufficiently large data source to be able to document what is the incidence of lung cancer by detail, race, ethnicity, gender, and smoking status, because we don't have smoking information within population-based cancer surveillance or cancer registry data. So this was a study co-led by my colleague Iona Chang and myself. This was funded through NCI. We used a multi-level integrative data approach. We combine electronic health data from two different healthcare systems, Sutter Health in Northern California, which covers about a third of the Northern California population, as well as data from Kaiser Permanente, Hawaii, which obviously is a much smaller healthcare system, but is enriched for populations of Asian American, Native Hawaiian, and Pacific Islanders. So we defined a cohort based on the dates of admission into the healthcare system, and ended up with a cohort of over 2.2 million members, among whom 1.275 were females. We extracted and harmonized EHR data for sociodemographic factors, as well as several known and putative risk factors. We geocoded and assigned a number of different geospatial measures, such as neighborhood socioeconomic status, or SCS, and ethnic enclave, as well as obviously air pollution is very important. So we incorporated information on particulate matter exposures and traffic density. We then linked these data on this virtual cohort to the respective state cancer registries of California and Hawaii to identify incidence, lung cancer cases, and extracted tumor specific factors and treatments from the cancer registry. So that was the approach that we used. And we needed to do this in order to get large enough numbers of distinct ethnic populations of Asian American, Native Hawaiian, Pacific Islanders for our analysis. We ended up with a population of almost 4,000 female lung cancer cases with using this approach. You can see here the breakdown of cases for distinct Asian American ethnic groups. We actually initially started out with looking at probably more than a dozen different single and multiple racial ethnic detailed Asian American groups. But these were the ones we ended up being able to look at in terms of statistical numbers. So I want to highlight for you here that, again, these are just among cases. This shows the distribution of smoking status. We're looking at never smokers or lung cancer among never smokers, LCI and INS, ever smokers, which is in green, and we did have some proportion of missing smoking data, about 9% missing data on smoking. So among Native Hawaiian Pacific Islanders, 15% were never smokers. Among Asians combined, Asian females combined with lung cancer, 50% were never smokers. These percentages largely match what we see in the literature. Among non-Hispanic white women diagnosed with lung cancer, 21% were never smokers, 14% among Black females with lung cancer, and 38% among Hispanic females with lung cancer. But let's look more closely now at the distinct Asian American, Native Hawaiian, Pacific Islander subgroups. We see 14% never smokers among Native Hawaiians, 20% among other Pacific Islander groups, 79% among Chinese American females. This is among females diagnosed with lung cancer, nearly 80% do not have a personal history of smoking. 53% among Filipinas and 58% among other Asians. This was published just about a month ago in JNCI. And we also looked at calculated incidence rates because we designed the study as a longitudinal cohort. So these are age-adjusted incidence rates of lung cancer for those same racial ethnic groups that's shown in the previous graph. You can see here the rate for non-Hispanic white women is around 10, for Hispanic women is around 8.5. And with the exception, interestingly, of Japanese American women, every Asian American, Native Hawaiian, Pacific Islander group had nearly 1.5 to 2 times higher rates of lung cancer compared to non-Hispanic white females. These are all female never smokers. And so this really is the first evidence we have of, in fact, a higher rate of lung cancer among Asian American, Native Hawaiian, Pacific Islander females who've never smoked relative to the general US population. We are wrapping up work on this R01. We are conducting attributable risk analyses in these 6 exposure domains. And we have my colleague Mindy Derawan has an administrative supplement to use the same data set to apply a machine learning, specifically iterative random forest approach to look at the joint contributions of multi-level factors to lung cancer and never smoker risk. And we've been fortunate to be able to follow up that study with our second R01. This is co-led by myself, Dr. Moon Chen from UC Davis, and Iona Ching from UCSF. And of course, Dr. Wakely is a contributor as well to this project. And this is funded by the National Institute for Minority Health and Health Disparities. We call this our FANS study, Female Asian Never Smokers. This is our amazing UCSF team, among whom we have bilingual, bicultural recruiters and study field staff. Our group at UCSF, again, led by Dr. Moon Chen and Clef Tepper, who is a geneticist. Dr. Wakely from Stanford, and we have, this is actually an early iteration. We have a much larger community advisory board now, but this is led by our CAB chair, Ms. Joyce Chen, who is of the San Francisco Chinatown Health Center. And really the effort is facilitated by Dr. Kim Rhodes, who leads our, who is our Associate Director for Community Outreach and Education and leads our CAB. So we have 2 aims for the FANS study. First is to identify the attributable risk of known, putative, and suspected multi-level risk factors. Not only are we focusing on multi-level risk factors, again, thinking back to that cells to society model, but we're also aiming to try to capture the cumulative exposures of these risk factors over the life course. With respect to etiology of lung cancer among Asian American female never smokers. Specifically, we're planning to focus on 3 exposure categories or 3 factors: genetic ancestry, individual level exposures, paying particular attention to culturally sort of specific exposures, and contextual level risk factors, including indoor, ambient air pollutants, social environment, as well as occupational context. And then our second aim is to characterize, we're using whole exome sequencing to characterize a mutational landscape and identify associated multi-level risk factors with different mutational profiles. We actually got this funded, believe it or not, as a population-based case control study. If there are any epidemiologists in the audience, you're probably in shock as we were. But we were able to make a compelling case that we had well-established methods to recruit representative controls, and that this really is one of the few ways that we can really robustly answer this research question. And in fact, I will add that this is the first study of lung cancer among Asian American females who've never smoked in the US. There's a number of studies being conducted in Asia. We are actually collaborating with some of our NCI colleagues from the intramural program of some of the Asian studies, but there's been no study done among Asian Americans in the US. So our goal is to recruit 600 cases using an early case ascertainment system from our cancer registry, which covers 9 Greater Bay Area counties, based on prior distributions of ethnicity among lung cancer cases. We expect about half to be Chinese, 100 Filipinas, and 90 Vietnamese. And then we will recruit matched controls, balancing across multiple sources. This is, again, a prior approach that we've developed and tested, shown to be effective in terms of recruiting a group of population-based controls. And so these are some of the sources that we're recruiting controls from. And we are still ongoing. We've been recruiting for about 6 months now. So hopefully next year or sometime I can come back and present some actual preliminary results from this study. And the second study I wanted to present, in which we've applied a multi-level approach to look at structural factors and interactions with omics, is our work on prostate cancer in African-American men. Why prostate cancer in African-American men? I think these are statistics that many of us are familiar with, and has been really persistent and ongoing. There is a higher burden of prostate cancer among African-American men. They have an 80% higher incidence rates and over twofold higher mortality rates. We also know that African-American men are more likely to be diagnosed with more aggressive tumors. However, adjusting for stage and treatment, some, but not all studies, some studies such as those conducted within a VA system, clinical trials, suggest that there may be comparable survival, but within population-based data, we see continuing disparities even after adjusting for these factors. So we thought about this particular disparity within this conceptual framework that you see here. We hypothesized that the higher rate of prostate cancer aggressiveness and mortality is ultimately driven by upstream factors, including that are shaped by life course stressors, particularly those that are social in nature, at the contextual level and experienced at the individual level. These contextual level factors that we hypothesized are structural racism and racial residential segregation, which in turn impact the social and built aspects of environments that men live in. At the individual level, this leads to experiences with discrimination, both major forms of discrimination, day-to-day or everyday discrimination, as well as adverse experiences, not only in childhood, but also over the life course. These together in turn produce stress, which are subsequently embodied and impact upon tumor biology and immune system, which in turn impact prostate cancer disease, aggressiveness, and mortality. And all of this is potentially moderated by prostate cancer risk loci as well as genetic ancestry. And in fact, there's some evidence to suggest that these contextual level or neighborhood level social stressors may be associated with prostate cancer. A study linking census data to cancer registry data in Pennsylvania show that social adversity was associated with shorter time to disease progression and a significant interaction between selected genotypes and neighborhood factors. As well as that socioeconomic deprivation is associated with Gleason score, which is an indicator of disease aggressiveness, neighborhood socioeconomic status, and individual education together and interactively were associated with prostate cancer risk. These last two were work done by our lab, and also were associated with prostate cancer survival and slightly explain the survival disparity between Black and white men. There's also biological evidence to suggest that stress embodiment may be associated with prostate cancer, both risk and progression. So these both in vitro and in vivo studies show that stress exposure increases development of prostate tumors. This is looking with mouse models, decreases effectiveness of hormone therapy, results in less programmed cell death and less clearing of tumor cells, and produces neuroendocrine signaling effects on growth metastasis, angiogenesis, and apoptosis, and that this occurs primarily through an immunological mechanism. So sort of putting all this preliminary data together, we collaborated with many collaborators and proposed a study called RESPOND. This is focusing on prostate cancer among African-American men, addressing the role of social stressors, genetics, and tumor genomics and biology. This is overall led by, so this is funded through what's called a U19. We actually proposed it as a PO1, a program project grant. It was ultimately funded as a U19 collaborative grant by the NCI as well as the National Institute of Minority Health and Health Disparities. We also have funding from the Prostate Cancer Foundation. This is led by Chris Heyman from USC overall, and it's split into 4 distinct projects. I am the lead for project 1, in addition with my team at UCSF. Chris Heyman is the lead for project 2. John Carpton and Franklin Huang are leads for Project 3, and Karen Spanos is the lead for Project 4. And I'll show you in a bit what those projects are focusing on. This is a national study. We are recruiting, our goal is to recruit 10,000 or so African-American men with prostate cancer, primarily from state-based cancer registries that are both SEER and or NACER cancer registries. And so you see some of the sites here, as well as sites of our collaborators. And so these are the 4 projects. So now overlaying the projects onto our conceptual framework. Within Project 1, we are looking at the life course social stressors. Within Project 2, led by Chris Heyman, he is going to be looking at the genetic ancestry and prostate cancer risk loci. Germline genetics. Project 3, led by John Carpton and Franklin Huang, is looking at the tumor genomics, and Project 4, led by Karen Spanos, is looking at tumor inflammation within the tumor microenvironment. And I should say that our goal ultimately is to put all of this together, again, thinking through back again to that South to Society framework, really trying to see the extent to which these factors interact together to impact prostate cancer aggressiveness and mortality. These are the aims for the project that we're leading at UCSF: examine associations between neighborhood social stressors and risk of aggressive prostate cancer. Aim 2 is look at exposures to multi-level social stressors across the life course and risk of aggressive prostate cancer. First aim uses geocoding and linking to cancer registry data. Aims 2 and 3 are going to be using the survey and genetics and genomics data that we are collecting among the 10,000, the cohort of 10,000 African-American men with prostate cancer. And then aim 3 is the one where we will be bringing together data from across the 4 projects to look at the associations between these multi-level social stressors over the life course and the genetic and genomic factors in association with aggressive prostate cancer. Within aim 1, we are collecting a range of data from using different approaches. We are collecting individual level data on the social stressors as well as other covariates. These are the life course multi-level social stressors that we are collecting. This is primarily done using both geocoding, linking to geospatial data, as well as through a survey that the men fill out, capturing neighborhood factors, lifetime and everyday discrimination, medical mistrust, adverse childhood experiences. And this, we've actually adapted the CDC scale to capture adverse experiences over different life periods, a form of active coping called John Henryism. This was a model developed by Sherman James, as well as different sociodemographic factors, family and personal health history, prostate cancer treatment, and lifestyle and behavioral factors. And through geocoding, but again, as well as also through surveys, we are collecting contextual level data on social stressors. Our primary factor, independent factor of interest, are 2 measures of structural racism. Through collaboration with Dr. Kirsten Beyer from the Medical College of Wisconsin, she has used Home Mortgage Lending Act to develop 2 measures of contemporary structural racism. One is a redlining index, and 2 is an index of racial bias in mortgage lending. We are also incorporating several different both local and MSA level measures of racial ethnic segregation, as well as looking at local measures of racial ethnic composition, using both a traditional measure of capturing percentage Black within a neighborhood, as well as racial ethnic typologies, combinations of different racial ethnic groups within neighborhoods. And then we're also looking at the potential mediating effects of different social and built environment factors, social environmental factors such as crime, socioeconomic status, built environment factors such as walkability and food environments. And I'm just going to show you briefly some preliminary data from our aim 1. So this is aim 1, again, is based on geocoding and linking these area level measures to cancer registry data from across the different response states. So these are the states included within respond, or for Michigan, we only actually are looking at data within Detroit. And I will just kind of orient you to this graph. So this is the measure of redlining developed by Kirsten Beyer using the Home Mortgage Lending Act data. This measure basically looks at the odds of denial of a home mortgage loan within a census tract relative to that odds of denial of a mortgage loan within the surrounding area of the census tract within a larger metropolitan statistical area. So it tells you if a particular area has a higher odds of mortgage loan denial than its surrounding area. And this odd is actually derived from a multivariable model in a spatial model, and it's adjusted for the amounts of the loan as well as the income of the loan applicants. So since this is an odd ratio, if the value is less than 1, so this is illustrated by the dark bars here, that means that that neighborhood has either equal or less odds of denial of a home loan. If it's greater than 1, and so that's all the lighter shaded bars above the dark bar, that means that that neighborhood has a greater odds of denial of a home loan. Okay. And so these we've basically mapped and assigned levels of redlining to non-Hispanic white prostate cancer cases and African-American prostate cancer cases within the cancer registry data. So what do we see here? Let's just focus on California. We see that African-American prostate cancer or men with prostate cancer are much more likely, so the lighter color bars, to live in neighborhoods where there's greater denial of a home loan, a mortgage loan, compared to non-Hispanic white men. And it's actually quite striking, the difference. Now, if we look across the respawn area, you see that this difference varies quite dramatically. For example, for Detroit, less than 10%, in fact, I think this number was somewhere around 5% of African-American men live in a neighborhood where there's equal or lower odds of denial of a home loan. The vast majority live in areas where there's greater odds of denial of a home loan. Okay. Now, this is a second measure of structural racism developed by Kirsten Beyer. This measure is capturing racial bias in mortgage lending. So now what this measure does is, again, takes the same data from the Home Mortgage Disclosure Act. It calculates for each neighborhood, each census tract, the odds of denial of a home loan for a Black applicant compared to a white applicant. Again, adjusted for the amount of the loan as well as the income. It's actually adjusted for income to home loan amount ratio. So what do we see here now? We see, again, if the value is less than 1, that means that African Americans have less denial of a home loan, equal to or less denial of a home loan compared to a white applicant in that particular neighborhood. If it's greater than 1, Blacks are more likely to be denied a home loan. When we first looked at these data, we were thinking, surely there's been some mistake. There's no way that these values, that the vast majority of neighborhoods have a greater denial of a home loan for Black versus white applicants. There was not a miscalculation error. This, in fact, was what the data showed. So not even focusing on the Black-white differences. Let's look across the states. Oops. And you could see that in California, for example, greater than 90% of neighborhoods have a higher rate of denial of a home loan for a Black applicant compared to white applicants. And for some of these neighborhoods, the odds ratios ranged from 15-fold greater denial of a home loan. Again, this is multivariable. It's adjusted for income to home loan ratio. And yes, as we would expect, Black men or Blacks are more likely to live in neighborhoods where their home loan was approved. So that's what you see in the data here. But this is certainly very striking and I think sobering as well. So we are continuing analyses. We are actually pretty far along with linking these measures of structural racism to looking at prostate cancer risk groups as well as mortality using the cancer registry data. To date, we've collected surveys on almost 7,000 African-American men with prostate cancer. So we're getting close to our goal of 10,000 men. We have a number of analyses also looking at the survey data. So I just want to close by offering some of these are sort of simmering thoughts that have been percolating in my brain in the 20 or so years that I've been doing this research. on sort of what I have seen the potential challenges but also opportunities are for us who do this kind of research. For one, I think it's important to always think about the framework and recognize that health inequities arise from fundamental causes and impact multi-level and also multi-level structural and social determinants of health. And so therefore, it's important we need to study these factors, these structural and institutional factors. There are actually fairly easy ways to do this without actually asking people about these very sensitive questions regarding discrimination. But you can, within our data, within the studies we conduct within our EHR systems, all you need is data on somebody's address. If you have an address, and no, not just zip codes are too broad and they don't align with census defined boundaries. But if you have an address or a census block group or a census tract number for where the person has lived, you can actually opens up a whole world of data you can potentially integrate in on neighborhood and structural context. But we can also take that a step further. We can also design studies in which we explicitly pay attention to capturing life course and cumulative effects of these types of exposures, as well as the weathering effects on both psychologically and biologically. Of course, as an epidemiologist, I have to speak to what our data needs are. Well, we need to start to collect, and we need to be consistently collecting structural and social determinants of health data. It's important, as you saw from our study with the Asian American, Native Hawaiian, Pacific Islander, lung cancer and never smoker female study, that the data be as granular as possible. It doesn't take that much more time to offer all the categories that one can potentially identify with or select. And in the end, you can always combine and pool based on the numbers. We recognize, so however, that there are sensitivities and concerns among those who collect these information, such as data that go into EHRs, the healthcare professionals, those in the front lines that are collecting these data, the admissions clerks. And so we need to develop collaboratively training and toolkits for how to collect these data in a sensitive and a responsible manner. We need to develop better methods and approaches for being able to conduct research within small populations. I think it's unfortunate that our current toolkit, which is based on having robust numbers of populations and sample sizes and study samples, restricts us from being able to conduct studies just because this population is small within our community, that we can conduct research within those populations. Consortia obviously is an approach for us to be able to pool data across multiple studies to get those robust numbers, as well as integrative data analysis, as the example that we showed you, where we combined EHR data from the Sutter healthcare system with Kaiser Permanente, Hawaii. And we also need both approaches, but also need to start addressing intersectionality interactions among multiple marginalized structural and social determinants of health statuses. And I will just end by putting up this graphic again, and to note that I think for us to really start to tackle health disparities and to ultimately achieve health equity, that we need to address the structural and social determinants of health, especially the upstream factors. Thank you so much for your time.
Thanks. That was fantastic. Really amazing overview of all this. So I've got a few questions I was thinking about as we were going through this, and I will mention to the audience that we do have the Q&A feature. So please feel free to type in a question, or I think you have access to the chat, and that would be great. So as we're thinking about, the inequities and sort of how those directly impact health. Obviously, there are a lot of different factors. And so with our work in lung cancer, we've looked a lot at air pollution and how that might impact things. And obviously, it's a little bit challenging to tease out the details there, but that can be helpful. One of the other big factors is diet, right? And accessibility to food. And I'm wondering how you're able to sort of weave that in. If there's like, what's the best way to sort of try to capture that type of information as we're looking at sort of that whole like global health and global from the global of that individual, right? You know, all of the factors that they encounter in their daily life that can impact their health.
Yeah, I think that's a diet is a great example of the multi-level, the relevance of the multi-level framework. I think there's certainly, you know, we often think about and certainly have been stymied by challenges within epidemiology of assessing diets using very detailed dietary food frequency questionnaires. But I think, as you pointed out, that it's important too to think about food availability and what one has access to. And when we think about a question like lung cancer among Asian American females, that context can change depending on where you are. It can change very dramatically if you are an immigrant from an Asian country, and even where within Asia you immigrated, and to where within the US you've settled and moved over time. You know, one can certainly ask individuals about what their typical diet is, what they consume. You can certainly get as granular as possible, but then you have recall problems. I know that dietary patterns have been an approach that people have started to look at, different types of diets and diet patterns. Within the food context, food environment, we've been using, our approach has been both asking folks about what they have available to them, but also using secondary data to capture that. We have been using information on availability and number of farmers markets. So within neighborhoods, we geocode, we get listings of farmers markets from farmer market associations, geocode them, map them to within neighborhoods of participants. And also using business data, we are able to capture some aspects of the food environment. But I think overlaid and interactive with all this is how is the degree to which ethnic types of foods may be available within it as well. And so that's a challenge that we're sort of trying to think about. How do we capture those within the geographical space?
Great. Right. Looks like Dr. Melissa Bondi, our chair of epidemiology and population health, has a question. Dr.
Hi.
Bondi. It looks like Dr. Gomez. Okay, there she is. Okay.
Hi, sorry.
Thanks,
Can you hear
Bondi.
me? Can you hear
Yes.
me?
Yes,
Hi.
we can.
Good. Scarlett, thank you for such an outstanding presentation, and I'm so glad that you were able to come and present this today. So first of all, your work is amazing, and I'm so lucky I've had the opportunity to work with you on the prostate cancer study, and after 2 years, it's nice to see that the sample size is increasing and that you've gotten to 7,000 cases, because I wasn't sure we'd ever get there, and maybe the pandemic was helpful to be able to reach people. So I was the person involved in the Texas site, and it was very challenging to try to recruit people in Texas. But, you know, just to actually reiterate some of these things, that being able to include, and I think this whole field of looking at social disparities is the ability now, and what Scarlett has done, and I just want to commend you, is being able to pull the data from all of these different sources, because epidemiology is so difficult. And I think the question you asked, Heather, about diet, you know, it's these kinds of things that I think in epi that have been a real problem. You know, people don't, it's the biases that we've had with confounding and other things when people report their diet. We don't know if that's true, but being able to pull sources of data through other data sources like Scarlett has been doing, really gives us a better estimate of social disparities and being able to have a better estimate of these things. And I feel like it's taking us to another level of really being able to understand structural racism. And the data you showed for the black-white differences in you know, with the home ownership and being able to get loans, be interesting to see if this is similar for other race ethnic groups as well. And I don't know if you've had the chance to look at that. Maybe, and also with the ovarian cancer studies that we're doing with ACEs and what you're doing with KP Rock, it's going to be really interesting to see if we see this in with gender differences. So I don't really have so much a question, but comments. And the one question is, you know, being able to look at this. I don't know if you had a chance to look at any of this with, probably not yet, with in the Asian American studies that you're doing, if you see the same kinds of things with home ownership and,
Yeah.
you know, does this crossover into other, I'm sure it does, but, you know, it just gives us a, you know, a chance to see how this really crosses over to all ethnic groups.
Yeah, that's a great question. And in fact, the data are there from the home.
Mhm.
I understand it from the Home Mortgage Disclosure Act data, that the denial data, the applicant data is available for broad racial ethnic categories. So theoretically, one could develop that, you know, that last measure that I showed, the racial bias measure,
Mhm.
racial ethnic groups relative to, for example, non-Hispanic whites. So I think that's definitely something on the horizon we're looking at, but I want to take the opportunity to kind of address that question, Melissa, from a different angle. Is that another aspect of neighborhoods that our group is really interested in is ethnic enclaves, and potentially sort of both beneficial but also detrimental effects of living in highly ethnic neighborhoods that are, you know, coined as being ethnic enclaves. That is, has more recent immigrants, may be linguistically isolated, and tends to maintain more of the cultural mores of a particular racial ethnic groups. And so we're using a number of different approaches to try to characterize these ethnic enclaves, both using census data, but also using other types of information to try to capture some of the potential social network and social support aspects of these ethnic neighborhoods. So I sort of feel like, you know, it can sort of go both ways. We did an interesting study where we looked at the more traditional measures of segregation, the ones developed by, you know, you've heard of terms of segregation like dissimilarity or blanking on what there's several different types of segregation that developed by demographers Massey and Denton. And so we looked at these measures in relation to breast cancer, stage of diagnosis and survival, and actually found, at least within California, though the findings were different in other states, that African-American women diagnosed with breast cancer, living in neighborhoods with greater segregation, actually had better survival if there were also more Blacks within that neighborhood, suggesting to us potentially a role of social support due to having greater co-ethnic neighbors. And perhaps that sort of social support mechanism arised out of necessity due to the segregation. So it was, you know, ecological data, observational data, but I think is provocative in terms of thinking about how potentially, you know, how ethnic groups distribute across neighborhoods may have both, you know, may over time potentially force residents to develop coping and social support effects. But it also forces us to think about what are the consequences biologically of having to constantly be on the alert and cope and, you know, develop these coping strategies.
So as I was listening to that, of course, was thinking about the work that we had done with, and Manali Patel had led, looking at the Hispanic paradox related to lung cancer. And so as I'm hearing about this data with breast cancer in the African-American community, I'm curious, as someone who's been very lung cancer focused as opposed to more broadly as an epidemiologist, if there are other examples of that, because again, it's that complexity of the whole milieu, right? So it's the support network, it's also air pollution, it's also food, it's sort of like all of those things interacting together. And in some ways, you know, as you talk about the housing challenges as regards to the African-American community and inability to get loans to have a home. And then we think about how these support networks can somewhat be helpful as well. It's complex. So I'm wondering if there are other examples too, where having that tight-knit community might actually be beneficial. So.
Yeah, absolutely. And I think that if we're talking, in terms of thinking about populations like Asian Americans and Hispanic Latinx populations within the US, there's the additional element of language. May come into play too with regards to living in more co-ethnic communities. So it's not only just potentially social support you derived from having more co-ethnic neighbors, also potentially more language specific resources. And we actually noted this in a qualitative study we did looking at where we were really curious to know, based on our cancer registry data, we were observing that for women with breast cancer, early stage breast cancer, for which there is an option, but really a recommendation for breast conserving surgery versus mastectomy, we were actually observing that Asian American women were more likely to have mastectomies when diagnosed with early stage breast cancer. And so we took a qualitative approach to try to identify what some of those factors might be, and noted that there were within San Francisco, Chinese women living within particular area of San Francisco, sort of Chinatown and surrounding areas. There was something unusual in that they were actually having a lot of breast conserving surgeries in contrast to their Asian counterparts living elsewhere. And so we took the opportunity of the qualitative format to try to inquire, well, why that is. And they all referred to they all went to one common hospital, so that was sort of one factor. But also, the fact that the community health centers within the area actually provided a lot of resources to facilitate them being able to travel to the hospital every day for a 6-week course of radiation. They provided the transportation, helped them to book the appointments, provided childcare, provided food if they needed it, elder care if they needed it. And so all of that supportive resources in language was in place. And so it made it easy for them to have a breast conserving surgery versus a mastectomy, which many women opt for because it's a one-time deal. So I thought that was an interesting lesson, and a vouch for the importance of qualitative data.
Absolutely. If we're not understanding exactly what all the factors are, it's hard to have the impacts we all want to have. So, well, great. I'm not actually seeing other questions, and I think in the Zoom universe, it's always nice when we get those 5 minutes in between meetings. So I just wanted to thank you so much for that amazing presentation and your willingness to have detailed conversation with us afterwards. So look forward to our continuing collaborations. And we've got a big thank you from Dr. Bondi in the chat. I'm not seeing others, so great. So thank you so much. And I know we'll be on a Zoom call again soon.
I'm sure we will. Thanks. Thanks, everyone.